More fallout for the reproducibility crisis, potentially affecting tens of thousands of studies. Oh, I love this. PCA-based findings in population genetic studies are highly biased and must be reevaluated. See, this is crazy because this happened not long ago. 2016, mind you, this is Scientific Reports, which is a nature-based paper, one of the biggest papers in one of the biggest academic publishing peer-reviewed journals you could be in. Science is right up there with Nature. 1 in 5 genetic papers contain errors linked to Microsoft Excel. What ends up happening is certain genes, expressions, if you type in certain expressions, they actually turn into dates. And because they turn into dates, they actually fucked up a bunch of analysis, apparently. The error often arose when gene names in a spreadsheet were automatically changed to calendar dates or numerical values. For example, one gene called septin2, commonly shortened to SEP2, is changed to 2 September and stored as 2 September 2016 in Excel. The researchers who published their data in Genome Biology say the issue can be fixed by formatting Excel columns as text or switching to Google Sheets where gene names are stored exactly as they're entered. This isn't just an issue that biology is new to. This is something that's been rampant in biology for a very long time. And the issue is when you're a biologist or a researching scientist who's in some sort of STEM arena. I was an academic. I was a neuroscientist. They don't teach you about databasing. They don't teach you about data storage techniques. They don't teach you about good data storage practices. They teach you, here's how you turn on the machine. This is how you collect data so it's as clean as it can be. Here's how you export the files. What the files are? Of the files that come out of any particular data collecting machine, the ones that the people care about are the CSVs. So like, here are the Excel files is what we were told. Here are the Excel files. These other things, we don't know what they do. Those other things are things like metadata of the machine's recording, machine printout stuff, regular scripts of like, here's just like a diagnostic of like what we did to collect the information. All those like good computer engineering, computer science stuff that you would typically find from something, some hardware that outputs something. All the stuff that you would typically open and log in a database of like, here are all the flags. Here's all like the metadata for the data set. And then the CSV, which is like, here's the actual data. They never teach you any of that stuff. They never teach you how to write in SQL. They don't teach you what Spark is. They don't teach you how to set up a database. They don't teach you any of that. So you typically have these gigantic CSVs and you load them all into MATLAB, and then you get like a script that was handed down from one generation to another. You don't know what the fuck goes on in it. You hit run and you see what happens. So it's like, that is the cutting edge of research right there in academia. I think one mistake based on technology, one is the inherent bias, but there's also some sort of bias in science, even though we pretend that science is blind. I mean, it depends what you mean by bias. Everyone makes assumptions. That's what a hypothesis is at the end of the day, right? Like, we get driven by falsifiability, and so you always have to make an assumption. But if you're talking about like just pure data collection, like naturalistic observation, uncontrolled, then that should be as blind or unrelated to any biases as possible. But even in the techniques that you use to record something, that could be biased, right? Even the languages we use to describe things, that's biased, right? PCA is a multivariate analysis that reduces the complexity of data sets while preserving data covariance. The outcome can be visualized on colorful scatter plots, ideally with only minimal loss of information. PCA applications implemented in a well-cited package, Eigensoft and Plink, are extensively used in the foremost analysis of population genetics and related fields. PCA outcomes are used to shape study design to use to shape, study, design, identify, and characterize individuals and populations, and draw historical and ethnobiological conclusions of origins, evolution, dispersion, and relatedness. Replicability crisis in science and products to evaluate whether PCA results are reliable, robust, and replicable. We analyzed 12 common test cases using intuitive color-based models alongside human population data. We demonstrate that PCA results can be artifacts of data and can easily be manipulated to generate desired outcomes. PCA adjustment also yield unfavorable outcomes in association studies. PCA may not be reliable, robust, or replicatable. As the field assumes, our findings raise concerns about the validity of results reported in the population genetics literature and related fields that place a disproportionate reliance on PCA outcomes and insights derived from them. We conclude that PCA may have a biasing role in genetic investigations, and that 32,000 to 216,000 studies should be reevaluated. That's a lot of dissertations. An alternative mixture population genetics model is discussed. When I was doing neuroscience, we did have a question of like PCA versus ICA. So there is a difference between principal component analysis and independent component analysis, right? Independent component analysis, unlike PCA, treats each component and tries to find linearly liking things, stereotypical signal in each of these components. And then it is up to the user to select which of these components are the ones that represent signal. So there's like a supervised part of ICA that requires some sort of expertise level, right? The other side of this was PCA, where in PCA you throw all the shit in and then it tries to, again, linearly create a component which combines like signal. Now the issue with PCA versus ICA, PCA, if you also have noise in there that is stereotypical, for instance, in neuroscience, if you are using EEG, eye blinking, which is muscles contracting and like pretty consistent, the EEG, the electroencephalogram, will pick up eye blinks as muscle artifact. So if you use PCA, you wouldn't be able to identify the muscle artifacts. So you would need to eliminate muscle artifacts before you did PCA, right? Whereas in ICA, for some reason, ICA would lump these stereotypical artifacts all together, and you could eliminate that component from your signal. So PCA typically would take like just the largest signals, put them together, see what happens, right? Overall, like you have like a supervised way of doing component analysis in ICA, a mostly unsupervised way of component analysis with PCA. You take assumptions from either and you go with it, right? In the case of PCA, they're finding that there is issues with PCA. They are biased towards all sorts of shit. Reliability concerns, validity concerns, disproportionate reliance on PCA outcomes. We're essentially lumping a bunch of data together and saying there's signal when really there might be no signal. We might be analyzing noise. And when you're talking about something at such a small, finite, tiny, literal molecular level, it's not like a game like baseball, or it's not like something like my face with muscles, where I could say, oh, wait, when you blink your eyes, this thing happens, right? When you're talking about this like extremely protein level, molecular level thing, it's hard to say whether or not you actually are looking at signal or noise. You have to be very confident that you're finding either, right? And as technology and statistics advance, so does the expertise in each of these fields, right? And that's from the molecular level, from the physics level all the way up. So this is definitely an issue because PCA isn't even meta anymore. Like, PCA is not the meta. PCA is a linear assumption, and when you have non-linear factors that you're PCAing, PCA fails. It fails to thread the linear because it fails to thread the needle because the needle is usually not a linear needle. So people have usually gone away from PCA and gone towards things like UMAP as their component or a clustering embedded layer technique. Just do random forest, bruh. Yeah. So PCA is not even meta. Like, UMAP's meta now. PCA is like, if you know everything in your data set is linear, then PCA works. But if there are some factors in there that are non-linear, then PCA is going to fail when it gets to your categorical variables or your ordinal variables, your nominal variables. Is going to fail there. Let alone like your scaled variables that have no linearity. We were talking about like multimodal data sets. It's going to fail there. So PCA is just, it's not meta. On the second part of it, even UMAP or like some sort of embedded layer from a neural net, that in itself also has its problems and assumptions, right? Like, not a lot of people like using UMAP or embedded layers unless you really have the exact problem to use it for, like thousands of features, where if you use an embedded layer or UMAP to reduce that down to, say, several features, like that's a speed up at the sacrifice of not a lot of accuracy, right? So you might use it in that case, but in actual like work practices where you only have like tens of features, maybe even upwards 100 features, a lot of people just throw the features into the fucking model because explainability these days is more important than speed and accuracy of model training. Whereas in the 90s, when PCA, ICA were like very new to STEM research, it was a benefit. In the 90s and the 2000s, it was a benefit because the machines, we weren't at the cloud computing stage yet. Machines did take a long time to run analyses. So I remember sitting like 2010 in an old lab, learning on like hand-me-down machines, being trained as a neuroscientist, working off like a fucking blue transparent top iMac. Like, the only improvement to this thing was like an increase in RAM, and I'm working off of this thing where we would run an analysis for literally like several days just to do one participant. And that's when we were like, okay, we can't do channel by channel analysis. What if we did ICA? And that helped us reduce a lot of the time. It went from days to a day, and that's still at a cost. One participant a day. So if you're running 30 participants, which is what our study had, that's a month just to pre-processing the data. So there was a time where component analysis was important because it helped with speed ups. But now my fucking, like, I could do all that shit in Colab faster than I could in my literal undergraduate lab where I learned everything. And it'd be not only faster, but most likely more accurate. Just packages these days have gotten not just better, faster, but more accurate, right? So, component analysis is sort of, it's not meta. It's not the way people typically go about things. In certain things, in certain arenas of data analysis, it is important, but in most arenas, it is not. Like, for example, I worked on a marketing team. You have literally thousands of demographic data, right? You have thousands of demographics from people. From one row, thousands of columns. Clustering methods, dimension reduction methods, embedded layer methods, they really helped figure out like clusters of people. It really did help. But that still required like sensitivity to knowing what these clusters mean, explainability in your clusters, what features were actually important to the project you were building out. You don't just do it to do it. You do it with the intention that it's supposed to help solve something, right? So sometimes it does help, but again, PCA is not the method we used, right? Because demographic features are not linear, right? Socioeconomic status, gender, race, religion, kids, education level, all that stuff is it's non-linear. And so PCA wouldn't be a good use in that data set, right? Just like how most things, I'm guessing, at the genetic level is also probably non-linear. PCA is something you learn in school and rarely used in work. That's right. That's right. Thoughts on forcing people to share their data and code for published papers in big journals? Feels like a lot of bad practice could be figured out with that. In my opinion, data sharing and open source should not just be a requirement of journals, but it should be a requirement for any study that is funded by some sort of federal entity, whether it is the NIH, the NSF, any school grant that school belongs to the public school system. I believe that it should all be open, and all the data collected should be open, because the people own that research. The people funded that work. Sure, there's research out there where it's like, you shouldn't make certain research fucking public if we're talking about matters of security issues. Sure. But when we're talking about like Nick Wayne's dissertation on people doing the prisoner's dilemma with EEG caps on, if that had federal money invested into it, that data should be open. Right? If it is below the level of security that is deemed by the government, then it should be available. It should be publicly available. Because here's a good example of why open source contributions are in the state that they are currently, and why academia has fallen behind. Open source contributions towards, whether it's any Python package you could name, whether it's scikit-learn, NumFocus related projects, PyMC, whatever the fuck, R related projects, all of the Tidyverse, all of the different sports analytics projects that we've worked on, any of these different scrapers that allow you access to data. Look at what that's gotten people, right? Look at what that has actually gotten people. It's gotten people actual access to open, not just data, but methods, right? All of it.